Speaker is Dr. Marianne Ashford from AstraZeneca, and she's going to present on the pharmaceutical development of nanomedicines, initial learnings and challenges.
Thank you very much, and thank you very much for the invitation. So I've been asked to talk a little bit about the pharmaceutical development and particularly the manufacturing of nanomedicines, and I'm going to do that by illustrating a couple of different types of nanosystems that we've worked on. So in this very brief talk, I'm going to give a very brief introduction and then, as I say, talk about the manufacturing side and some of the learnings, and some of these things are quite easy retrospectively, and some of the analytical characterization and a few reflections on some of the other things that are involved in nanomedicine development. So the two main systems, the two systems that we're really working on taking into development are the I'm not sure this is working. The Acuren platform on the left, which is X-bind therapeutics. So that is a polymer matrix with the drug encapsulated within the polymer and then a PEG stealth layer. So it's a PLA PEG polymer, about 100 nanometers in size, and we have a product there in phase 2 clinical trials. The second system is very different from a nano perspective. It's a lot smaller. It's a dendrimer-based system. The drug is attached by conjugation, something like 10 nanometers in size, and we've got that in preclinical development at the moment. And both of these has really been employed to try and improve the therapeutic index of small molecules. And this is coming from a background of a company that's used to developing small molecules in a standard range of dosage forms. So what's different with nanomedicines? So normally we might fix our final formulation after our phase 1, phase 2 studies prior to phase 3 and the pivotal studies. But with nanomedicines, we're having to do that a lot earlier. That's firstly because the nanomedicine changes the safety and efficacy of the products. We need that both for clinical trials and for the to going through tox. Also, we don't really understand our CQAs at this point, our critical quality attributes. So by fixing things early, much like you do in oral drug development for a BCS4 compound, we minimise risk. But that means we have to front load our CMC costs. So these projects tend to be a lot more expensive than standard development projects, because we may not know whether the drug that we're encapsulating has actually got proof of principle in the clinic. So normally you see major CMC costs at this stage after you know that. We know that the manufacturing process needs to be robust, flexible, and we need to be able to scale it up. It's important because these are non-biological complex drugs, so the control often comes from a very well-defined manufacturing process. The analytical characterization is absolutely key to make sure we have quality, safety, and efficacy of the product. And then we need careful consideration of the biopharmaceutic strategy, i.e. those factors that are affecting the safety and efficacy of the medicines, so changing sites, different batch-to-batch variation, and bridging between different studies. So a little bit about the manufacturing. Firstly, these are very, very different. This gives my formulation colleagues challenges, and this gives my chemical college challenges. So this has a relatively easy formulation, but chemistry is different. So I'm just going into this in a little bit more detail. So we're giving these to a chemical development department that are used to making drugs of 500 molecular weight. These are 100-odd kilodaltons, so it's very, very different. So there's a nice star structure and some stylized imaging, but probably it looks something more like this, or if it has been described as tumbleweed. So a dendrimer is a lot more structured than other polymers, but the majority of it is a polylysine core. But we've got PEG 32 PEG 2000 chains, trying to get pure PEG. And actually trying to make sure that the specifications as we think it is, so you don't get cross-linking of those chains, it has been a key challenge. Another challenge I'd like to say, and I learned afterwards, is the drug. So the drug we use is BCL2, BCL-XL inhibitor. Those who know about some oncology, they're cell death inhibitors. They have therapeutic index challenges. But this drug was already 20 stages to make it. So shoving a dendrimer process on the end wasn't very bright. So the head of chemical development told me after we'd got some really nice preclinical data with it. So that's perhaps a key learning, or just think about what you're trying to put onto or into these systems. So we have quite a complex drug process, which is quite long anyway. That's nothing to do with the nanotechnology, but it's worth thinking about in future. We then put the linker on the drug, and then we've got 13 steps to make the dendrimer scaffold, so adding the generations of the dendrimers, adding the MPEG, and making sure that we understand exactly what's coming at each of these stages, and we've got some specifications in each of the intermediates. What we are able to get is a common multi-program intermediate there that we can then use across a number of programs. So that's some benefits. We've now managed to make 8 kilogram batches at GMP GMP quality internally. So another issue for our colleagues is, as I say, we're used to small molecules, so we're used to using crystallization to purify compounds. So obviously you can't do that with dendrimers. So we've had to establish a non-aqueous GMP ultrafiltration, diafiltration to support that clinical manufacture. It might be quite obvious, but I think it's important to think about how are we going to get the purity that we think that we might need. And so it's just different technology, different techniques, different skills. So once we've made the dendrimer, it's a relatively simple process to make it into a product. We have to use non-aqueous because we've got a hydrolytically linked compound, simple filtration, and then it is filled into vials. One thing that's important, though, because it is a hydrolytic linker, is to make sure that we actually control the amount of free drug. So the important thing is to make sure at the end of the infusion into the patient, we have whatever we put in our specification, no more than that percentage free drug. We have to make sure we've actually qualified that within our early toxicity studies. So we have then got to work back very, very early on in the program and say, well, how much can we manage on manufacturing storage? How much can we manage on reconstitution? Do we have to have Usain Bolt sprinting to the wards to get it to the patients? What is that whole supply chain looking like? And we've done some nice mathematical modelling to make sure we understand exactly what's happening and the effect of temperature and pH. So just on the other polymer, the other nano we've worked with, and I'm just going to mention this very, very briefly because a colleague of mine, Marianne, has got a poster number 109 on this. But here it is for the formulators. This is a struggle. So here, this is what we might do for a solution, simple aqueous solution and sterilize. We're now looking at about an 8-stage process with a number of input and outputs and some complex input and output and relationships to really make sure that we have a robust process. And a lot more work to be done for our process engineers to really understand this. So very briefly, I wanted to, in the last few minutes, mention a little bit about analytical characterization, because I think hopefully I've shown you that there are different challenges in terms of manufacturing, but similarly for analytical characterization. So we need to make sure that we can properly analyze our nanomedicines, in that we can identify and characterize them. We understand the critical quality attributes, and some of that I think we're limited by some of the technology limitations, and we're certainly having to get to build our skill base and buy new kit. A little bit of a concern, analysts like measuring things, but they've got to measure the right things and the relevant things. And then otherwise you get loads of resource on the project just because the analysts fancied to get this nice nano with their bit of kit. And that doesn't help us in the industry to have the next one because everyone says how expensive they are. So yeah, it's a different learning, but it's an important learning. Just on sizing, this may be, those of you that are involved in size techniques is very obvious. This is a dendrimer and a couple of buffers, but each of these different sizing techniques gives different answers. It's hardly surprising because they're all looking at different things here. But the majority of what we did, and there's nice reviews out, most of the industry look at DLS, and DLS is fine, and maybe that's fine for qualitative changes. But really, we now need orthogonal techniques. We need to understand what's going on. And for different size nanos and different materials, different techniques are relevant and applicable. So as well as size as a critical quality attribute, affecting aggregation and distribution, the stealth layer is important for understanding PK, PD, and safety. Also is the release rate of the drug from any nanomedicine, and that free drug, which I've really mentioned a little bit in terms of making sure you control it through the life of the product. So very quickly, and it's hard to perhaps summarize all this in 10 minutes, a few further reflections. There is probably a lack of development and regulatory precedents going through to the product stage, and all nanoparticles are very different, so that not all the learning can be taken across the different systems. There is, however, help from both the NCL and the EU NCL, and I would urge anybody to look at all their protocols and look at what's been written. There's also new guidance available back end of last year from the FDA, and there's a workshop in a couple of weeks on that new guidance. I think I've alluded to also mentioning we need novel advanced analytical techniques. That requires new equipment, new capital, new funding and capability and new skills for people working on these projects, and it's often very different to what they've worked on before. We also need, and I haven't really gone into this in this brief talk, bespoke materials, and to make sure that we've got a robust and reproducible supply chain, and that we understand the specifications. And if you've got, less than 0.2% of a diacid in the PEG, is it really less than 0.2%? And what is your molecular weight of your polymer? Because it does make a difference. It is also challenging to measure release drug in vivo. It's part of the guidance, it's important, and it doesn't help us in pharmaceutical development and understanding batch differences and understanding what the CQAs are and for bridging. And I think that's a real challenge and a bit of a gap in the field, because I don't think we've sussed it properly for all nanotechnologies. And we're also not only changing the approach in pharmaceutical departments, but we're changing them across multidisciplinary departments in the industry. And maybe because it's come in from a pharmaceutical department, it's struggling to get your toxicology college and DMPK college and just make sure everybody's on board and understands what we're doing. So finally, just to say, it is different to making tablets and simple injections. It starts earlier. The manufacturing is more complex. It needs to be very well controlled. We require that advanced characterization and capability build. I'd say mathematical modelling has been really useful for us, and I'd urge a lot of people to think about using and adopting it early. It's important to collaborate, and I think it's important to be proactive, engaging with regulatory guidelines and attributes. And before finishing, I just want to acknowledge all my many colleagues that have worked in this field with me, our 0466 project team. I showed some of that data, and external collaborators in Xbind Therapeutics and Star Pharma. Thank you. Apologies for being late.
Thank you.